Biotechnology Part 2. So in this section, we're going to focus on genetic engineering and what it is, the formation of transgenic organisms, primarily focusing on genetic modification of organisms. We're going to look at the role of plasmids or bacteria in this process of genetic modification. We'll look at a process that allows scientists to make a lot of copies of particular sections of DNA or particular genes so that they can be studied or modified. And in this section, we'll wrap up with cloning. So genetic engineering or genetic modification is altering an organism at the level of the DNA. So this isn't like splicing. This isn't the production of a hybrid of a tree by grafting another tree. This isn't, and I've seen this before, and you probably have seen it too, where there's this lab rat and it's got a human ear on its back. It's just being used as a place to grow a human ear. Itself is not producing human ears. So genetic modification is actually altering the DNA. Now, we'll look at the different results of these, but looking at how this happens, there can be several different ways that this happens. One of them is through gene silencing, which is referred to as a gene knockout. And that was first done with mice by switching off or turning off or forcing a gene not to be expressed and kind of looking at the ripple effects. So what would be the positive benefit of doing that? And then what could be some of the ripple effects that might be negative from doing that? So that was first done with mice. And then introducing a gene from the same species, and you could see that in something like gene therapy, or even inserting or introducing a gene from a different species, which is called transgenesis, which results in transgenic organisms. So transgenic organisms have their DNA manipulated somehow to express a foreign gene. So once chromosomes or genes have been recognized, what they do, what they code for, there are enzymes that can be used to cut them. They're called restriction enzymes, and I mentioned yesterday they're very specific to what they cut. And you can cut pieces of DNA to leave kind of a hangover of a couple of bases that can then be joined with DNA of another organism. So two different ways that DNA can be cut. It can be cut in a blunt end that's straight across, or it can be cut in a sticky end in which it can combine with DNA of another species or of the same species. But basically, at the genetic level, it can be joined with any species because DNA is universal. So everybody's A goes with a T, and everybody's G goes with a C. So the word transgenic, again, breaking everything down, this is already in your notes. Transfer or across, so trans is across, that's how we put it in our notes. And genic, of course, is referring to genes. So transferring genes from one species to another. Transgenic organisms are referred to as genetically modified. But not all genetically modified organisms are transgenic. Some GMOs, though, contain DNA from two different species. So when you have that DNA from two different species, when it's combined together, that's called recombinant DNA. So here you see an example of a transgenic species. All right, so I'm just going to read this with you. Transgenic goats have been modified to produce spider silk in their milk. So some people call these spider goats, but that doesn't mean that they have 8 legs What it means is that they've been modified in such a way that they have genes from spiders that produce that silk they make their web from, which is very strong. They have been joined with the genes of the goat. So in the goat's milk, it produces this substance that becomes the silk. It has to go through a processing, but this is something that could be very beneficial. So that silk from spiders, that's used in things like parachute cords. So this could be a quicker, cheaper way to produce those in mass quantity is through spider goat, spider silk milk. This is disgusting, but we're not going to drink it. We're going to process it and we're going to make parachute cords out of it. Okay, so I found some examples of some genetically modified foods. So the first one there is the golden rice. You can see the golden rice on the left, and then the natural white rice on the right. Flavor Saver tomatoes. So on the left, the GMO, and on the right, the normal one. That's the rotten one. BT corn. So the genetic modified one looks good. The normal one's starting to look yucky. Aqua Advantage salmon. So the genetically modified one is the big boy in the background, and the normal one is the little tiny one in the foreground, and then glow in the dark animals. And I don't think I need to show you which one that is. So why? Why in the world would you want to do any of these things? Well, we'll look at it for just a second. So the golden rice. Rice has been modified to produce vitamins. Now, rice we know is a staple for a lot of the world. It is a food staple. So if you found a way, and they have, through this recombinant DNA of daffodil genes and rice genes to produce this new rice that is fortified, it has created naturally vitamin A, then you can have a lot of the world that is healthier and not suffering from malnutrition. Flavor Saver tomatoes. So the Flavor Saver tomatoes came about in the 90s, which seems like forever ago. You all didn't even experience the 90s, but these have since gone by the wayside. So they came and they've went. But what they were known for is that they lasted longer. They spoiled more slowly. But the name Flavor Saver is kind of misleading because the reason they were trashed is that they really didn't taste good. So what's the point? BT corn. So the BT corn is modified with a bacterial insecticide genes. So instead of having to hose down your corn crop to keep the bugs off of it, this corn is going to produce its own insecticide. Now, this is something that's kind of went back and forth over the years. Several years ago, Taco Bell was using a genetically modified corn product and its tortillas, and people were having allergic reactions to it. Not to the corn, but to the bacterial genes and what they produced as this natural insecticide. But that one's been trashed. The Aqua Advantage salmon, that big boy at the back, is going to grow faster. And you think about it, you could have a food that's readily available and more of it. And then the last one, the glow in the dark animals. Well, there's no apparent reason for this, obviously, but there have been glow in the dark cats, glow in the dark puppies, glow in the dark pigs. The first that I ever saw in one of my very first textbooks at the school, like 20 years ago, was a glow in the dark tobacco plant that had genes from a lightning bug. So the gene from the lightning bug, what caused it to luminesce was a gene. Well, it resulted in luciferase. I don't know why I was going to say transferase. Luciferase. Okay, so I'm going to pause here You pause your video and we'll come back together, but we're going to watch this video on a transgenic organism. Well, it's going to show you several, but the main focus here and the one I want you to look at is this cow that they're going to genetically modify. And they also mention a little bit about a pig as well. So I'll see you on the other side of the video.